Hello everyone, myself Vivesh Kapoor and I work as a scientist at the Birbal Sahni Institute of Paleosciences, Lucknow. Today I will give you a glimpse of preparatory and related works on fossil vertebrates that we undertake in the Vertebrate Paleontology and Preparation Laboratory of our institute. The Vertebrate Paleontology and Preparation Laboratory at Birbal Sahni Institute of Paleosciences was inaugurated on the 26th February 2018 with an aim to prepare and study fossil specimens of vertebrates. Vertebrates are animals that possess a backbone or a vertebral column, and they represent subphylum Vertebrata. Examples are fishes, reptiles, amphibians, birds and mammals. And vertebrate paleontology is the branch of science concerned with fossil animals having a backbone. In the field, the vertebrate fossils can be found in articulation or disarticulated remains. Generally, it is the latter scenario. Thus, careful preparation of these fossils is required both in the field as well as in the laboratory. In the field, we explore for these fossils in various sedimentary sequences. Once we discover vertebrate remains, we note information such as the fossil locality, coordinates of the locality with the help of a GPS, lithology that yields fossils, the stratigraphic setup in the field. We also note as much as information regarding the settings or plans in which the various parts of the animal lay in an outcrop. We provide field catalog numbers to individual specimens. Many a times, it becomes necessary to stabilize the specimens using various chemicals such as a mixture of polyvinyl acetate and acetone before retrieval. At times, we encase the entire blocks of rock that show embedded fossil remains by preparing a plaster of Paris jacket before retrieval. We also collect rock samples in the field for retrieving associated microvertebrate fossils in the laboratory. The large size skeletal remains are carefully packed like in wooden boxes with foam linings on the inside to sustain long journeys to the laboratory from field locations. In the laboratory, the POP jackets are carefully removed and we initiate the process of preparation with the help of pneumatic air scribes. The pneumatic air scribes work with the help of compressed air. For this, we use an electrically powered 100% oil-free air compressor unit. The compressor unit has a horizontal 90-liter capacity tank, a 2-horsepower motor that generates an airflow at a rate of about 10 CFM with a maximum pressure of 121 psi. The compressor is attached to an automatic timer-based drain valves with oil and dust filters. The compressed air inlet in the laboratory is attached to a pressure regulator that can handle pressures up to 140 psi. The air pressure regulator is attached to an industry standard pneumatic air scribe that generally works between 100 and 120 psi pressure. We should note that the air scribes come in various types and sizes along with various types and sizes of bushings and styluses. Various combinations of these equipments are used in the laboratory so that we can remove both hard and soft matrices from the fossilized bones. The pneumatic air scribes work like a micro jackhammer, and the preparation works on fossils are undertaken using portable magnifiers or optivisors under dedicated light sources. The magnifiers allow us to observe specimens up to 10x magnification during preparation. As we remove the matrix, we carefully use various grades of stabilizers and adhesives to treat fragile, cracked or broken fossil specimens. The prepared specimens are photographed using high-end digital camera and measured using vernier calipers, manual as well as digital, and a goniometer. We also prepare molds and casts of the specimens in the laboratory using plaster of Paris as well as using industry standard hardeners, resins and pigments. The rock samples associated with skeletal remains are also macerated using various chemicals. The slurry of individual samples are separately sieved using ASTM standard sieves. Later, the sieved sample are observed under a stereoscopic microscope for retrieving microvertebrate fossil remains. For photography and measurements of the microvertebrate remains, we use a stereoscopic microscope attached to a camera, which is in turn attached to a computer. Various softwares installed in the computer allow us to photograph as well as digitally measure the microvertebrate fossils up to 8x magnification. The morphometric information of the prepared specimens help in vertebrate paleontological research in the context of estimation of body masses or sizes of fossil vertebrates, inferring their taxonomic and evolutionary status, inferring patterns of global distribution of vertebrates in the geological past, inferring the community structure, and many related aspects. It is important to mention here that the laboratory environment is exposed to various types of hazards that include fumes from various chemicals, noise, and dust. Thus, as per health and safety guidelines, the laboratory personnel are encouraged to use protective equipment of international standards that include gloves, dust masks, ear defenders, and eye protectors while undertaking preparatory works on fossil vertebrates. Thank you for watching and hope you enjoyed the module.
Hello everyone, welcome to the Vertebrate Paleontology and Preparation Laboratory at BSIP, Lucknow. Let me show you the lab inside. Here we prepare vertebrate specimens which we collect from faraway places in remote areas from India in different sedimentary regimes. As you have already seen in the presentation, all the specimens are retrieved manually, prepared initially for retrieval in the field, and then they are packed in these boxes here. And these boxes are shipped from the field areas and they come here. So all these boxes contain skeletal material of different vertebrates. So if I take a block from this side, it should look almost like this. This is almost a 60 to 70 kilograms of block, and you can see the bone fragment here embedded in the matrix. Apart from that, we are, as I already told you, we have jacketed this. This is a skull fragment, so we have jacketed it for protection. After this process, we use this pneumatic airstrip here, which is controlled by this pressure regulator here. And you can see the pressure regulator here. It will increase the pressure. Right now it's 0. And the scribe works like a micro jackhammer, and we can remove these skeletal remains from the matrix. We individually remove these specimen remains, and then this is how the specimen which we get comes out to be. But this is not prepared. You can see the sediment which is already attached to the bone fragment. This is the bone fragment. These are the sediments, if you can see closely. After that, we use some stabilizers and glues, and after preparation we use some stabilizers, some glues, and we come out with the whole fossil like this. You can clearly see this was not a single this was a single specimen, but clearly broken at many places. So this has been joined and we have a complete specimen now. Similarly, you can see all the block I showed you earlier. We were able to retrieve all these bone fragments, which are now neatly prepared and glued and stabilized for further studies. After that, we try to measure them with a goniometer. This gives you the angle which we need to measure on different bones, and a vernier caliper here. This is a mechanical one, but we can use digital ones as well. These are for measuring the bone thicknesses and different measurements, the lengths and all those things, width, lengths, all those linear measurements. Apart from that, we try to prepare casts of different bones or skeletal material. This cast is made up of POP, that is plaster of Paris, and this is from a hardener and resin. With the pigment used, you can see the pigment here is grey, and here we have used a green pigment, and you can clearly see the fossil. These are the. Apart from that, the rock matrix which comes from once the major bones megavertebrates have been retrieved from the rock fragment, we also use the rock itself and macerate this rock using various chemicals, and we sieve that slurry of the material, and then after sieving we dry the sample, and the sieved material looks like somehow like this. These are the various standard sieves which we have, and you can have the you can have the sizes in microns as well as the different standards there are. Then we bring this the macerated material, and we use these trays here. You can see that these trays are of different colors. One is a white checkered and one is a black checkered brass plate. So these are picking trays, and with the help of different types, with the help of needles and different sizes of brushes, we try to retrieve the sample from here. And after that, the specimens which we retrieve of microvertebrates, we save them or we store them in these slides. There are different types of slides. This is a 40 chambered black based 40 chambered slide. But there are white based as well as 24 chambered and different sizes, round punch slides as well. So you may use different types depending upon your requirement. After that, I showed you that we measure the larger fossils using vernier calipers. It's not possible for micro vertebrates to be measured via vernier caliper. So we use this high-end stereoscopic microscope. And this is the eyepiece of the microscope, and this is the focusing knobs, and this is the magnification adjuster here. It is attached to a camera, a high-end digital camera, and this camera is attached to the computer. We use various softwares because we are using a Leica model here for the microscope. We have a Leica software, Leica application LAS 4.8 version, and this software helps you to measure the fossil as well as to photograph it digitally. So if I show you one particular sample here which we have retrieved from the same sediments. We can do some adjustments here to see as per the scale which we are using or as per the magnification we are using. So if you want to, you can focus this. There is a slight difference between the focus under a microscope and on the screenshot. So you have to adjust the focusing when you are visualizing the specimen using a software. So you can take these measurements here. I can show you how. You have a scale here. So if I want to see the length of or the height of this tooth, it's a fish tooth. So you can clearly see how much micrometer this particular tooth's height is. It is 0.684 mm is the height. You can take other measurements. Similarly, for other particular fossils, you can move the scale anywhere and you can take any particular dimension you are interested in. And similarly, you can use it for different types of fossils or different types of fossil remains in invertebrates. So I'll show you a skull here. This is the whole skull of a mammal which we have retrieved from the block which I already showed you here. And this is the airstrike. We prepare this I need to use a dust mask. This is the scribe we are using here. We can control the pressure of the air on the scribe as well. Apart from the pressure regulator, you can switch it off here with a knob, or you can switch it on, increase the pressure as per your requirement. This is the skull of a mammal which we have retrieved from that particular thing, and I'll like to show you how we prepare it. For preparation, we need to be very we have to work under a magnifier. This is a portable magnifier with LED lights. These LED lights can be switched from a bright white light to a bright red color light. You can see here, this is a red. So depending upon what particular type of light you require, you can adjust this light arrangement as well. So you bring it close. This is almost like under 10x what we are working. And it works like a micro jack, and you can see that I am able to remove the sediment. So I try to remove here. This job requires a lot of patience. Although we are using a portable LED magnifier here of high magnification, we can also use some headgear like a magnifying optimizer here. It goes onto the head like this, and it has a 2.5x magnification, and you can use it. It comes in different magnifications as well. You can use it like this, and you can work closely on a specimen. Like if you want to work on this specimen here, I can literally use this visor. I can bring it close, and I can work on the scribe like this. As already told you, this job requires a lot of patience, and one specimen can take up to few days to few months. Like this skull, we have taken almost 4 to 5 months to prepare this particular skull fragment. As the environment here is in the lab is a bit dusty, so we use protections like an eye protection and dust masks, which are P100 standard, and ear defenders, because of the noise level, to protect the noise level. Thank you so much.